Could someone explain why is there controversy about using embryonic stem cells?
If you ain't got no money, 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 if you're independent, get your money, girl. 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 Everybody in the club right now, tell the DJ to turn it loud. Dedicate their favorite song. Single ladies, you can't go wrong. Everybody in the club right now, tell the DJ to turn it loud. Dedicate their favorite song.
Embryonic stem cells are obtained from early-stage embryo, a group of cells that forms when a woman's egg is fertilized with a man's sperm. Extracting stem cells from embryos raises significant ethical questions.
As scientists are discovering new ways to treat human diseases, many problems have arisen. Embryonic stem cell research is one of them, and its relevant ethical problems are what we are going to discuss. First, to know who have taken the survey, the following information is going to show us the demography. The first problem that comes to embryonic stem cell research is that it destroys human embryos. So the question leads to whether embryo is an individual person. This is what people think. To me, I think when somebody can feel, I would consider him a person. Here is a comment about if embryo is a person or not from the survey. Another point that could be looked at is whether independent individual is more important than an embryo. The point there is whether we can consider sacrificing embryos for children or adults. Since I believe cells without sensitivity are not human yet, I believe independent human lives are more important.
Why? Where do these embryos come from?
The embryos being used in embryonic stem cell research come from eggs that were fertilized at in vitro fertilization clinics but never implanted in a woman's uterus because they were no longer wanted or needed. The excess embryos were frozen and later voluntarily donated for research purposes. The stem cells can live and grow in special solutions in test tubes or petri dishes in laboratories.
Oh, I see, but why can't researchers use adult stem cells instead?
While research into adult stem cells is promising and moving forward rapidly, adult stem cells may not be as versatile and durable as embryonic stem cells are. Adult stem cells may not be able to be manipulated to produce all cell types, which limits how they can be used to treat diseases, and they don't seem to have the same ability to multiply that embryonic stem cells do. They're also more likely to contain abnormalities due to environmental hazards such as toxins or from errors occurred by the cells during replication. However, researchers have found that adult stem cells are more adaptable than was initially suspected. There have been significant advances in work with adult stem cells.
Wait, what are stem cell lines and why do researchers want to use them?
Line is a group of cells that all descend from a single original stem cell. Cells in a stem cell line keep growing but don't differentiate into specialized cells. Ideally, they remain free of genetic defects and continue to create more stem cells. Clusters of cells that can be taken from a stem cell line and frozen for storage or shared with other researchers. That way, researchers don't have to get stem cells from an embryo itself.
How about stem cell therapy? And how does it work? Stem cell therapy is also known as regenerative medicine. It is the replacement of deceased, dysfunctional, or injured cells with stem cells or their derivatives. It's somewhat similar to the organ transplant process, but uses cells instead of organs. Have stem cells already been used to treat diseases?
Big Mega Radio Smasher.
I had the time of my life, and I never felt this way before. And I swear this is true, and I owe it all to you.
Oh, I had the time of my life.
Oh, yes!
Stem cell transplants, also known as bone marrow transplants, have been performed in the United States since the late 1960s. These transplants use adult stem cells. Adult stem cells are being tested in other applications, including a number of degenerative diseases such as heart failure. Stem cells from umbilical cord blood have been successfully used in clinical trials to treat cancer and blood-related diseases. The Food and Drug Administration has approved an initial safety study using an embryonic stem cell therapy for the treatment of serious spinal cord injuries.
How about therapeutic cloning? What benefits might it offer?
Therapeutic cloning is a technique to create versatile stem cells independent of fertilized eggs. In this technique, the nucleus, which contains the genetic material, is removed from an unfertilized egg. The nucleus is also removed from a somatic cell of a donor. This donor nucleus is then injected into the egg, replacing the nucleus that was removed, a process called nuclear transfer. The egg is allowed to divide and soon forms a blastocyst. This creates a line of stem cells that is genetically identical to the donors. In essence, a clone. This technique is also called somatic cell nuclear transfer. Some researchers believe that stem cells derived from therapeutic cloning may offer benefits over those from fertilized eggs because they are less likely to be rejected once transplanted back into the donor. And they may allow researchers to see exactly how a disease develops.
Has therapeutic cloning in people been successful?
Researchers haven't been able to successfully perform therapeutic cloning with humans, despite success in a number of other species.